Good morning, everyone. Yeah, so I'm Aurélie Vinet from herein, and this work was carried on with colleagues from herein and from IDEL. Yeah. So at first, as I was a bit surprised about how results at first, I will provide you some a brief non-exhaustive overview of some results obtained in the bibliography. There is a problem? No. Oh, sorry. And then I will begin with the methodology we used, and sorry for repetition with the presentation of Mario this morning, but I will be quickly. Then at last, how result of the evolution of the trade-off between fertility and production under different climatic conditions from France, and how result of GWAS on heat tolerance traits with a focus on the trade-off between traits. No. Yeah. So to estimate the genetic correlations between production and fertility, the field data are always used for cattle. And the choice of the traits is very important because each of them have the advantages or its own advantages and its own disadvantages. And here are some examples of production traits that have been already used for this kind of analysis. So in dairy cattle, and for example, the 305-day yields or cumulative yields for a shorter period, for example, for the first 60 or the first 100 days. We can use also the stage day production performance, and usually we have only one performance per month for this records. And we can use only one performance, the closest to the insemination or the closest to a given days in milk, or we can use several performances during a given period. And some authors also estimate the genetic correlations between production and fertility using persistency as a production trait with a different method to estimate this persistency. Yeah, for production, many studies focus on the first lactation because from the second lactation onwards, the less fertile females and the low yielding ones are usually no longer present. So it can bias the analysis. So the results of this genetic correlation are close. Results of the correlation are estimated with milk yield, protein yield, or fat yield for as a production trait. So it doesn't change anything to consider one trait or another one, or little change. Yeah, so as previously said, each trait has its drawbacks or at least its limitation. For example, the 305 days production usually does not take into account the effect of the gestation. And this can penalize the most fertile female because from the fourth month of the gestation, there is a downward effect of the gestation on the production performances. I've put it here how results of the effect of the gestation length here on the milk yield per day. And you can see, for example, that here. And this result are obtained in the two breeds we studied, in blue in Holstein and in brown in Montbéliard. And you can see here that between the 6th and the 7th month of the gestation, there is a drop in production of around 7% in how primiparous Holstein. And this, if you don't take into account the gestation, it can mainly penalize the most fertile females, those that can be pregnant at the beginning, at the early period of the lactation. Well, we can use also TSD performances, either only one single performances, but in this case, it provides less information about the dairy potential. Or you can use several or all TSD performances, but in this case, it could be better if you take into account the interactions between genetics and tasted milk. Because the interactions between genetics and the stage of lactation can be quite high. And if the genetic variants of the production evolve along the tasted milk gradient, there's a good chance that the genetic covariance with the other traits evolve too. So, and if we take into account this, such interactions between genetics and tasted milk, the models, it's a bit harder to run. Yeah, now for the co-fertility traits, there are basically three kinds of performances: intervals, counts, and all or nothing traits. For the intervals, you can have, for example, the interval between the calving and the first service, named the days to first service, or the number of days open, the interval between the two calvings, or the interval between the first service and the conception. For counts, the trait is usually the number of services, and you can have all or none traits, either for the first service or for all services. And in this case, the most commonly used are the non-return rate at 56 days or the conception rate. And when the conception rate focuses only on the first service, is usually named the first service conception. Well, and you can see that the limitation of all these traits is that at least one insemination must be recorded to obtain these traits. And this excludes cows that are the least able to manage a trade-off between lactation and reproduction, or the least fertile females, and in particular those that don't ovulate. Yeah, the problem with the intervals and the counts is that they may depend on the farmer's choice. For example, the first service of high-yielding cows can be voluntarily delayed to give them more chance for a successful insemination. Well, yeah, if the beginning of the lactation, at the beginning of the lactation, the cow produces a lot, sometimes breeders prefer delay this the first insemination. And another example is that after an successful insemination, the chance of a new service are higher for high-yielding cows than for low-yielding cows. Okay? And this last point may also affect the all or nothing traits when the all services are considered. It can be based by the number of observations at the total. The number of observation can be based by this choice. So it seems that all or none traits at first service are the least biased by the farmer's choice, right? They are the less biased, but there are also binary traits with no repeated data. And sometimes it can be a bit hard to catch the genetic variability with such as binary and highly complex traits, because fertility is always a high complex traits. Particularly with field data, because we are the result of a long process of biological processes. Yeah, and despite this drawback of the binary trait and no repeated data, this is a choice. We decide to work on this trait, conception rate at first insemination, the same trait as Mario just presented with the local study. So, and don't worry about this boring table. I just want to show you some some ideas. Here are some examples of the genetic correlations between different production traits and all unknown traits. And all these results are obtained in Holstein cows or crosses with mainly part of Holstein. And all this, in all these traits, the fertility trait is the result of the success at the first insemination only, except in Salt and collaborator, where all services are studied. And so the fertility trait is the first service conception or the non-return rate. Well, and yeah, here, each of these studies provide the results at the first insemination, among others, and some of them consider the lactations, the different lactation as repeated measures. So we have a global result and not a result at the first insemination. So yeah, so here are in red are the most negative genetic correlations, and the result goes up to positive correlation in green. Yeah, so it's difficult to draw a clear trend from these results, but it seems that the most negative genetic correlation at the top here it was obtained when the beginning of the production traits is only at the beginning of the lactation, when the energy balance is the most negative, and when it's harder to deal simultaneously production and fertility. So the most the cows with the most yielding, the most important male yield are those who have the less chance to successful first insemination. Yeah, but be careful. But this study is a bit old, so the cows are very different today. But yeah. Okay, if we look at the other negative correlations, they are mainly obtained, and they are all obtained for the worst them, when the trait considered is the 300 days in milk, but the results remain highly variable. When the tasty performances are considered, the variability of the result is weaker, with results from slightly negative to almost zero, which is completely different. Yeah. And when the persistency is used, the results are a bit contradictory. Sorry. Well, however, when these same studies consider not an all or nothing traits for fertility, but an interval tip or a count tip traits, the results are more stable toward longer intervals and higher counts for the high yielding cows. Why I showed you all these results, this boring table, it's because estimating the genetic correlation between production and fertility seems to be highly sensible to the traits, to the models, and perhaps also to the population studied. I don't go to the question about the evolution of the genetic correlations or not during the time, but the population 30 years ago, 40 years ago are clearly not the same, even in Holstein are the population today. So, and for instance, the worst sires for fertility, even they're good for production, they are no longer used. So even more since genomic breeding values. Yes, so so we need to be aware of the pros and the cons of the traits to date and the traits used, and we must be careful about the conclusions that can be done from our estimates. Well, here are the complete reference if you want to see some of them more in more details. Yeah, so briefly, and one of the objectives of the Rumigen project was to not only estimate this trade-off between production and fertility, but also, and above all, to find out how it would evolve with rising temperature. And the underlying question was, is this trade-off can be amplified by its stress? Yeah, a few words about the methodology we used. So I don't spend time on this. Yeah, I just want to more time on the G by E models, where when you account for interactions between genotypes and environment, you allow you estimated breeding values to evolve across along a gradient, an environmental gradient. And here the gradient is a THI. It's exactly the same THI as Mario presented this morning. And you can have an evolution of the estimated breeding values for an animal. And here there are 3 animals. And so you can have re-rankings between animals for a trait according to the THI considered. And if the estimated breeding values can evolve, so the genetic variances, covariances, and the genetic correlations too. Yeah, and as the THI can be described as a continuous variable, we could use random regression models with Legendre polynomials. So in this model, the performance of the animal I here is not only related to the pedigree and other phase effects, so not only related to the pedigree, but also to the THI at which this performance was recorded. And this can be done thanks to the Z matrix. So this matrix here you can see, yeah. And yeah, this Z matrix is composed of vectors that depends on the THI. And here I've put the first 4 Legendre polynomials, how they evolve between -1 and +1, and because it's a range of values for the orthogonality of these polynomials. And well, yeah, I'll see more later. So in blue, you have the order 0. This order doesn't depend on the THI. It provides intercept, and it gives how much the cow deviates from the population mean over all THI, over all environmental conditions. In orange, the order 1 is a slope that gives how much the cow deviates from at extreme THI from other cows. And the following the next order, the quadratic function and the cubic functions are no real biological meaning, but they give flexibility to the curve. Yeah, so yeah, just it's another point that explains the importance of the standardization of the environment. When your covariate is a cubic one, the covariate can be very high. So your genetic coefficient of regression is very, very, very low, and it's a bit hard to manage with such difference between values of each genetic coefficient of regressions. Well, for example, if your THI range from 20 to 80, in your vector Z at THI 50, you will find this value for the 0, for the 1, 2, and 3. It's very easy to build the Z matrix. Well, as when you fit your genetic value with 4 polynomials, then you have to estimate 4 genetic coefficients of regressions. Here are the genetic coefficients of regressions. And these genetic terms are not dependent on the THI. They do not depend on the THI. To obtain the estimated breeding value at a given THI, at your chosen THI, you have to multiply this vector of genetic terms by the Z vector corresponding to the THI you chosen. Okay, and if you have 4 genetic terms, the genetic matrix is like that. So it's a 4-4 genetic matrix with here the variance for the intercept, the variance for the slope, and so on, and here the covariance between slope and intercept. And once again, this matrix doesn't depend on the THI. And to have the genetic variance at a given THI, you only have to pre- and post-multiplied your G matrix by the corresponding vector Z at a given THI. It's a bit hard to obtain the results, the estimates of this matrix. But once you have this matrix, it's very easy to estimate all the genetic parameters. And you can do the same for estimate the covariance for a same trait at 2 different THI, but we have to consider the appropriate Z vector. So the vector at THI, 50, and the transpose of the vector of the THI at THI 70. And with the appropriate variance and covariances, you can have the genetic correlation between the same trait at 2 different THI. So it's exactly what Mario presented this morning. So be careful. The more regression functions there are, the more the genetic variance will be partitioned. And the model is not necessarily better when there are a lot of function of regressions. For example, in our G by E analysis, most of the additive genetic variance was explained by the first component, by the intercept. Usually 80% of the genetic variance and sometimes 90% of the genetic variance is explained by this term. So the remaining genetic variance has to be split, have to be partitioned by the other terms. So it works well when your genetic variance is very large, but when you have very weak genetic variance, it can be a little tricky. And sometimes it's better when you reduce the number of polynomials. Well, and here are some examples of results we obtain with such models. Here are the evolution of the genetic variances as a function of the THI. Here are for the Montbéliard, but the results in Holstein are similar with the same trends. On the left are the results for the protein yield, and you can see that the genetic variance decreases at high THI for the protein yield. And this trend is the same for all production traits, the protein, fat, milk yield, and the same for the contents, protein and fat contents. So for production, the genetic variance tend to decrease at high THI, and the trend is the same for the heritability. And on the right, you can see the result for the functional traits, the somatic cell score, and the conception rate. And in the opposite, in this case, for functional traits, the genetic variance tend to be higher at high THI. And once again, the trend for the heritability is the same. We have higher, for fertility, it's a bit, the difference is very weak because we go from 2% to 4%, but the heritability is higher at high THI. So, and here are the genetic correlation between the same trait at 2 different THI. And here, maybe with the mouse it's maybe clear, but I'm not too pole. Here you can see, for example, for the conception rate at the left and the protein yield at the right, you can see here the genetic correlations between the conception rate at THI 50 and the conception rate at THI 70. And the two graphs are symmetric. And you can see that here we have also the genetic correlations are given by the color. For blue, it's a genetic correlation equal to 1. And for the green results, this corresponds to genetic correlation equal to 0.9. So you can see, and it is the case for all the traits we studied, that there are very few interactions between genetics and THI in our data. And we obtain exactly what Mario said this morning. The genetic correlations for a same trait at different THI is very higher for production traits than for fertility traits. But in general, in our data, the genetic correlations are high. So we have very few or no genetic interactions between genotypes and THI in our data. So we have fewer rankings between animals, fewer rankings, but we have scales effects, particularly due to the evolution of the heritability and the genetic variances. For the protein yield, the difference between animals and between the worst and best, but the complete variability, the difference between the animals for protein yield is weaker, is lower at high THI. Whereas for the functional traits such as somatic cell score or conception rate, the trend is to an increase of the difference between animals at high THI. And to conclude with this methodological aspects, I want to define the traits I will use in the following slides. You can see here the evolution of the estimated breeding values as a function of the THI for the protein yield and for the conception rate on the right for conception rate. And here are some results for the Montbéliard breed. These are the top 10 and flop 10 for these two traits, top and flop within traits. Oh yeah, so this such curve allow to determine the genetic values, estimated breeding values for all the sires at THI 50 for the protein yield here, the breeding values at THI 70 for the protein yield, and the same for conception rate. And in the following slides, I will name this trait the level. Level for production for protein yield at THI 50, for protein yield at THI 70, and so on. And we can define another kind of traits, the slope. The slope gives the evolution of the ranking at high THI for us. We consider only the slope at high THI. Here, the THI 70, the high THI for our population. So the slope provides the evolution of the ranking, so the trend to be more or less affected by its stress. And this slope, to obtain the level, it's a genetic value, so you have to multiply the genetic terms by the Z vector as the appropriate THI, 50 or 70. And for the slope, it's almost the same, but you have to multiply the genetic term by the derivative of the Z vector at, not at the THI, the Z vector at the THI considered. You have to derive with respect to the THI. Well, I can show you this evolution of EBV without reminding you that as Mario presented this morning, for all kind of traits, for all performances, the THI produce a decrease in performances. So here are EBVs. So they give the ranking between animals at each THI, but it's important to consider the results within RTHI. And phenotypically, all the animals experience a decline in performance. Yeah, and here it's an illustration with the prediction of protein yield or reception rate. So it's the same. Here's the estimated bonic values, and here's the prediction of the performance. You can see that, for example, some animals lose less than others. And this is what is reflected by the slope. It's only to compare the animal for the the loss more or less impacted by its stress. Well, at last, a result of the evolution of the trade-off of the genetic correlation between production and fertility under different climatic conditions. Yeah, for this study, we used the field data from the French national database. We focused on the first lactation performances and on performances recorded between 2010 and 2020. For production, we use the protein yield traits. These are 30-day performance records, so we have only one performance per month. And we have restricted them to the period 80 to 200 days in milk to avoid taking into account the interaction between genetics, and lactation stage. And for fertility, we use the conception rate at first insemination, and we only retain the inseminations performed between 50 and 180 days after calving, because it's a period where most of the first service records, the first service are performed for both breeds and in general for cattle in France. So this study involves 3 million of Holstein cows and 600,000 Montbéliard cows. It's okay. Well, the meteorological data are were provided by the National Agency, Météo France, which estimates daily meteorological variables for each square. It's not very clear, but these are square of 8 kilometers by 8 kilometers. So we have data for each of these square. And this there is a model under these values, and each weather station helps other to estimate the values. And we use the THI exactly the same as Mario presented to me this morning, which is mainly dependent of the mean temperature, the average temperature, and slightly modulated by the relative humidity.
So you pick the farm closer to the center of the.
Oh yeah, no, it's a good question. Now you choose the data because to estimate the genetic components, you have to build a really beautiful data set. Now you choose the data, and we have the postal code of the Earth, and we have I averaged for each city, for each village, I averaged the meteorological data according each scale and so on. So I can rely the THI in one commune with the herd of this commune.
It's good you have this, particularly for your country, because by having this, you don't have problem in terms of if you base the decision on distance, you may have a mountain just here.
Yeah, but
by this, they extrapolate that. So it's like they are correcting my altitude and everything when they give you the temperature they give.
They take into account the altitude, but the problem is that I don't know where the cattle is at a specific day. And we have the postal code, the zip code of the herd, but I don't know precisely where the cows are. And usually, but no, but usually for dairy cattle, they are close to the farm. It's not the case for beef cattle, but for dairy cattle, usually. But yeah, I don't know precise. I haven't the really precise value. Yeah.
Is this the prediction for the plus 8x8 feeds? Is it done regularly or it was also deployed?
It's done regularly. Météo France estimates, I think it's each day, and they can re-estimate data of the previous day according to the data obtained today.
I think you can get
And-
some parameters as well.
I never compare. The great interest is that we can have this data for all squares, and when we want, we can have data for not the newest one because the re-evaluation of the results, but for instance, I load data. No, I think. Yeah, I load data. We can load up to the last month, and it's free for French researcher.
Free.
Totally free for French researcher. Well, two different THI were used. The average THI over a 3-day period before the test date, including the test date for the production. And for fertility, it was the average THI over a 8-day period after the services. This THI were chosen because these are the THI that allowed to estimate highest effects on the performances, respectively. And you can see here on the graph the distribution of the THI associated with the performance records. So in blue, again, Holstein, in brown, the Montbéliard. For conception rate, the results are with a solid line, and the dashed line is for the protein yield THI. The average THI is around 60 for both breeds and traits. Yeah, and we have only few data recorded at THI 50. It's also kind of trade-off between high THI and sufficient data, enough data to make statistics and to estimate good estimates of the genetic component. If the data sets are too scarce, the estimates will be wrong, clearly. So it's another kind of trade-off to decide because it can be hard to know when the cows are on its stress, because the variability and so on and so on. And here we are quite sure that the stress has begun for dairy cattle population. For France, it's clearly not the case for Spain, but for France, yeah, it's a very hard THI for us.
It is not what? It is not the case in Spain, not because it's not the case because, of course, at that temperature we have mitigation systems, but
Yeah.
you don't have.
No, you don't have.
So, I mean, in terms of if we didn't have those systems, for sure that would be.
Yeah.
I mean, the threshold is below 70 for sure.
Yeah, we don't have most of them
You will end up with those.
don't have elevated system, and moreover, most of the dairy cattle populations are in the northern part of France, so the coldest one. Yeah, so the models are bivariate reaction models using Legendre polynomials, and as the conception rate has very low genetic variance, very large data sets are required for an accurate estimation of the variance and covariances components. And so we opt for Sayers models that make it possible to handle very large data sets spanning all THI conditions with a relatively reduced pedigree. And as only one and moreover, only one performance per echo can be used because we studied the conception rate at first insemination. And the Sayers models are appropriate for describing the effect of the THI gradient. Because we have only one performance at the THI for echoes, but we have many performance of the daughters of the Sayers at different THI. And we can span all the THI gradients. Well, so for these are the models. So the model contains fixed effects and fixed effects specific to each trait. And here, the genetic effect, the Sayer effect, was modeled with 4 Legendre polynomials for the production and with only 3 Legendre polynomials for the fertility. And here you can see the genetic variance obtained. You can see here the part for the protein yield terms, here the part for the fertility, the conception rate, genetic coefficient of regression. Here. So for example, here, the variance for the intercept for the prediction, the variance for the intercept for the conception rate, any grade, and it's symmetric. So if it's symmetric, maybe, no, there is a problem here. No, here. There is a problem here. Sorry, forget this part. Here, the part dedicated to the covariance between the protein yield terms and the fertility terms. For instance, here the covariance between the intercept of the protein and the intercept of the conception rate, and here between the intercept of the protein and the slope of the fertility, and so on. And when you see the size of this matrix, maybe you can understand why I don't want to take into account another kind of interaction between genetics and days in milk. If I consider this interaction, I have to add other coefficient of regressions, and it's very hard to estimate such genetic terms. So that's why I prefer to focus on the middle of the lactation. And once again, quickly, you can estimate, for example, the covariance between the level of the protein yield at a specific THI and the level of the conception rate at another THI. So we have 2 different traits and 2 different THI. We can use, in this case, the G part dedicated to the covariance between protein and fertility terms, and multiplied by the corresponding Z vector. Z vector for the protein yield at THI 50 and the vector for the fertility at THI 70. And well, thanks to the appropriate variances and covariances, you can estimate the genetic covariance between the level of protein yield at THI 50 and the level of conception rate at THI 70. As an example, you can do everything you want. And you can do the same with the slope. And if you use the slope, you don't use the Z vector, but the derivative of the Z vector. It's quite simple. For protein yield and for protein yield, we have several performance per co. So we had a non-environmental permanent effect of the co, and this effect was modeled as the same than the Sayer effect. And the residuals. Well, we consider the residual heterogeneous across THI classes. And we don't exactly as proposed this morning. So in the middle, we choose a similar interval by 10 THI. And at the beginning and at the end, we consider classes that that can allow to have sufficient data. But estimated the residual variance is not the most complicated, but we prefer the an equilibrium between places. Yeah, I think it's better, or I advise you to consider heterogeneous variances for residual variances, because if you consider it homogeneous, you can see here with this simple example. If you consider this variance homogeneous, you constrain this genetic variance to evolve as the same manner than the phenotypic variance. If the data here increase with THI, and this one is constant, you may force this one to increase with the THI. So maybe it's right, but at the beginning, we don't know how this variance is evolved. So maybe it's a good idea to just check how they evolve along the THI. Yeah, here are the results of the genetic correlations. Yeah, no, it's time. The evolution of the genetic correlations between the conception rate and the protein yield along the THI gradient, and considering here the same THI for protein and fertility. The results, once again in blue for Holstein and in brown for the Montbéliard. And here, THI 50 is the average situation, the average THI. So we can consider that it is average situation for selection today. So we are close to the genetic correlation between these two traits today. And so the result for the genetic correlation is around 0.14 for the 0.14 for the Holstein, and close to 0 for the Montbéliard. And when we go to a stress situation at THI 70, the genetic correlations go to 0 or almost 0 for Holstein, and drop to minus 0.16 for Montbéliard. Yeah, and the standard errors are not so bad. Well, so yeah, all results for the Montbéliard are skull case. So perfect illustration of what we thought at the beginning, but the reality for Holstein seems different. So yeah, and as you can see, regardless of the THI, yeah, this correlation are rather weak, from 0 to minus 0.2. And we thought that one of the main reasons that explains this result is because we focused on the milk lactation. We exclude the part with when the energy balance is the most negative. Clearly, I have to check that. It can be very interesting to perform the same kind of analysis with the protein yield at the beginning of the lactation. But yeah, the result is also this one for when we consider the milk lactation. And for us, it's the choice seems logically, because it is a period when most of the inseminations were performed for France. So there is a period where the cow has to manage similarly, simultaneously, production and reproduction. Well, if you look at the relationship, I think there was a question this morning about that. But here are the relationship between, the relationship between the levels at THI 50. Here are the graphs I presented you before, with the estimated breeding values for the conception rate and for the protein yield. So if you calculate the genetic correlations between the level at THI 15, so approximately the current situation, the current breeding values of the animals, and the trend of the breeding values in warmer conditions, so with a slope at THI 70, you can see that first, for conception rate in Holstein, this correlation is 0, clearly. So we have the same kind of results as Mario presented with a slope independent of the intercept. When we here the result in Montbéliard, it's the results are a bit different because in Montbéliard the genetic correlation for conception rate is positive in our population. This means that the cows with here the worst breeding values, the most negative breeding values, are in general those with negative slopes, which means that the cows that today or already experience high difficulties in reproductions will have even more difficulties at its stress situation. So the positive view is that the best cow for reproduction will be the less effective effect in its stress situation, but yeah. But well, once again, for Holstein, we found a genetic correlation equal to 0. So we can't use this method to select better animals for future. And for protein yield, this genetic correlation is negative for both breeds, meaning that the animals with the best breeding values for production will be the most affected with negative slopes at high THI. Yeah, and this best animals for production with the best EBV are also those are the most affected for production and also for conception rate. And I give you here the values we obtain between the levels for the protein yield and the slope for conception rate. Is that okay? So,
and that's even that I remember this result. Actually stronger in Montbéliard than Boston.
Yeah, always. Yes, higher. This one. For this one, we have very high standard errors. Not for Holstein, but for Montbéliard, we have a little less data. Yeah. Sorry, I have to continue quickly. And what about the genetic correlations between the two slopes, the slope of decay of the conception rate and the slope of decay of the protein yield? These correlations are positive, but very moderate. So the good news is that these slopes are not antagonistic. Well, I don't know if it's a good news, but I have no time to explain that. Sorry, a bit long for the first part. And so clearly, the most important thing is that clearly these slopes are not the same heat tolerance traits. And they do not necessarily select the same physiological processes. And this is a question we try to answer with our GWAS analysis. So we have breeding values estimated. Sorry, it's very quick, but I have to be fast. We have breeding values estimated at all THI using random regression model, and this provides information about the tolerance through traits such as the comparison between levels at 2 different THI, the slope of the degrees of performance at each trait situation, and we add for this GWAS another trait, the difference between the two levels. And we have done that for fertility, production, and health traits. And yeah, so the aim of this work was to understand the genetic determinism of heat tolerance and the source of the variability. to learn about the role of the variants that could be selected with an tolerance index, and probably the most important for the course on trade-off, to identify variants with opposite effects depending on the traits, because I remind you that for production, levels and slopes are negatively correlated. Yeah, so the data are quite similar, but here we study the milk yield, and we had the somatic cell score as a health trait, and both are from test day performance recalls. The fertility trait is the same as before. We have genomic data, 50K microarray data for all sire insemination sires, and some complete data for the 1K genome project that allow to impute the 50K genotype at the sequence level. The methodological data are the same as previously described. For this analysis, we first run a blood animal model, which involved 5 million of Holstein cows and 1 million of Montbéliard. Blood animal model with random regressions and so on and so on. You well know the methodology in herein. Well, in this model, so the performance is the sum of the mean, the fixed effect of the variance, and each of the first these are within breed association studies. So Holstein in one part and Montbéliard in another part. And so we tested the association of the 13 millions of variants one at a time. And we have a random polygenic effect to catch the familial in the familial informations, and a residual. Yeah, one of the drawbacks of this such random regression models and such traits we analyze is that we have no phenotypes, we have no performance of levels, particularly with how data. It can be different with automatic milking recording, but with only one performance per month, we don't have any performance for levels at THI 50, levels at THI 70, or for slopes, even more for slopes. So these are not measured traits, but predicted traits. And so we have to use EBV, and more precisely the regress proof from EBV, and we weighted each performances by the equivalent record contributions. And as the reliability of the regress proofs are higher for sires, we implemented how GWAS on the sires populations. And to ensure the same linkage disequilibrium for all analysis, we focused the analysis on the size that have all the performances, all the DRP, after data editing, according to the reliability, to the fact that each sires have to add at least one cow, one daughter with a phenotype that particular THI, and so on and so on. So finally, we have 4,600 Holstein sires and 1,700 Montbéliard sires. And yeah, no, it's not interesting for us, the imputation. Yeah, let's go to the results. First, we found no difference between the QTL associated with the level at THI 50 and the level at THI 70. It's within traits. Within, I can show you, I think, yeah, I have the graphs. Here are the result of the GWAS with, on the axis, the 13 millions of variants along the 29 autosome. And here with the y-axis, the p-value, minus log of the p-value. And you can see here, it's example for Holstein and for milk yield at two different THI. But it's exactly the same trend for all breeds and all traits, that the QTL are exactly the same. And this result is not surprising because in our data, we found very few interactions between genotypes and environment. So the ranking of the animal is approximately the same, whatever the THI. So we found no region of the genome specifically associated with the THI at its stress situation and not with the traits at some kind of comfort situation. However, these traits, these GWAS are still of interest for detecting QTL that can be antagonistic between levels and slope. So we found some QTL of intolerance. So yeah, some of them are already been pointed at as intolerance regions. Just no time to details, but I just to show you VPS 13b. VPS 13b we found variants in these genes that are statistically associated with the slope of decay of the milk yield. And this variant, this gene, sorry, has already found in Chirouillou and collaborators in 2021 with the same kind of traits in Australian population, in Australian Holstein. Yeah, and we found also new QTL for intolerance and so on. Yeah, another interesting result is that how QTL of intolerance for slope or difference of level, this QTL of intolerance are all different from the QTL from the levels. All but VPS 13b. If you see it here, result of the GWAS for Holstein for the level at THI 50 for milk yield and the slope of decay of the milk at THI 70. I don't give you the exact position, but I check. And I can't give you if you want, but the same variant, I think the responsible variant is T. The same variant has a positive effect on levels and a negative effect on the slope. Sorry, because as the effect is expressed in the unit of the trait, it could be better if I put it in a standard deviation. Sorry, because we can't compare directly the level against the slope. Okay, and the QTL of intolerance are all different according to the performance analysis. It was also a very interesting results. Yeah, here are the results for Holstein. You can see here the result for the three slope. It's slope of the degrees of performance of milk yield, degrees of performance of somatic score, and of conception rate. And you can see that we found no common QTL. No. So, wow. Yeah, first, it's consistent with how polygenic result because we don't know the genetic correlations between these traits, but we know the genetic correlations between protein yield, not milk yield, but protein yield and fertility. And clearly the slopes, yeah, the correlation exists, but it's very moderate, 0.2, 0.3. So clearly it's not the same trait. And we have the proof here that the QTL detected are not the same. So the slope of degrees probably have a part of intolerance, but clearly it's not the unique function that can be selected with this trait. And maybe some other function can be hidden on the... Clara, you have a question? Yeah, so it's just an idea, an hypothesis, and clearly I don't know, but maybe different slopes can be hidden, a kind of ability to prioritize a function rather another one during stress. But I don't know. We have a lot of work to perform. So in France, the trade-off between production and fertility is moderate, and it's when we consider the male lactation production only. And the trade-off remains more or less stable in each stress situation. First, we have few performance recorded at THI 70. Yeah, it's clear. Another point is that the technicians told us that when the outside temperature, the outdoor temperature is high, usually the performance recording or the inseminations are postponed to milder days because they have low performances this day or a few chance of success insemination, so they prefer postpone this record. So yeah, okay. So sometimes, we have days at THI 70, but usually we don't have performance associated with this day. And another point is that how genetic by THI correlations are high, and we have few genetic, few interactions. And first, as Mario said this morning, probably this such model underestimates the interactions. And moreover, I am quite sure that farmers try everything to avoid heat stress and try to alleviate the heat stress for the animals. For example, for the crops, you can do anything. Usually you can do anything to alleviate heat stress, to alleviate drought stress. And but for breeders, it's possible to alleviate heat stress, to provide water, to provide shade, or to change the feeding, the hour of the feeding, and so on. So because plant genetics found a lot of G/E for climate, but for animal, it's usually hard. So I think it can be an explanation. So quickly, however, it's important to look at this trait because animals with the best breeding values for production today will be the most affected by temperature increases, both in terms of fertility and in terms of production. Yeah, so unfortunately, we found few QTLs of intolerance traits, and probably because we have weak irritability and not measured but predicted traits. And but it seems that we found some genomic regions that could be selected for intolerance without detrimental effect of levels. But clearly, we have a lot of analysis to confirm this and a lot of work for intolerance, select animals for intolerance. Well, I have no time to thank all colleagues and partners for Wageningen University and Research and INIA and IFTA, but some of them are here today. So thank you for the very interesting discussion during this project. I'm sorry I'm late.
Questions for Aurélie?
Going to implement this, are you going to include this in the immunogenetic?
Yeah, there are two problems, at least. First, the irritability of the slopes are very low, but in a genomic area, it's not a problem. But no, the irritability are low, but our main problem is that we need more information, I think. For example, the correlation with the longevity, because we are pretty sure that select on the slope of the degrees of the conception rate, it's a good idea, because it's if you want to maintain a herd, you have to reproduce. So yeah, but we are not all we have not the same idea in the tip. But I wonder, even if the correlation is positive between the two slopes, I wonder if it's really a good idea to maintain high production for high yielding codes. Because we know that high yielding produce a lot of internal heat, and it's clearly a problem when the outside temperature is high. They can dissipate the extra heat. So I'm not totally convinced that it's a good idea.
The problem is not the problem is not a problem with genetics. The problem is a problem with farmers, that they are very reluctant to give up production. Very reluctant. So they think that they put fans and springboards and everything that will be there. And I don't think they will. I mean, they are. I think dairy cows are reaching a physiological.
Yeah, you're right. The problem with animal breeders is that they can allele it. And when we see, for example, for plants, there are more progress for plants because they can't allele it. And there are more progress for plants than for animals. Today, for cattle, I can't speak about the other productions. Yeah, because they found a solution. But finally, we will have problems with fertility, health.
Yeah,
We are sure.
It's a matter of time.
Final? Yeah.
Now, the question regarding your model, if you model the polynomial functions for the rational, and I say that the QHI values for your input in the model is from 20 to 70. Like from 20 to 70.
Yeah. Yeah.
Yeah, but to my understanding, like for keep stress is maybe above 65 something.
Yeah, but for France, en fait, sorry, it's my French. For France, we saw a decrease in performance for milk yield and for all production traits at 50-55. Clearly lower than in Spain, or maybe lower than from the Netherlands. Yeah, because we have a lot of animals with posters for France from spring to. I think it's one of the reasons.
I also wondering, like, if the speed stress starts from 55, like you said, maybe you can just your option, like you use the phenotypes from 50 to 450 or above 50, and you can just feed a linear reaction on. And that be a solution for detecting this.
No, but we impose a restriction that you have to have a number of records up in after 50. Don't you remember?
Yeah.
And we edited the data.
First, we are not sure, and clearly that the THI, the threshold THI for us, the threshold from the beginning of the degrees, we are not sure that this threshold is the moment of the beginning of the stress, because there are a lot of things that change in the environment, particularly because the animals are outside and go to pastures, so the feeding change and so on and so on. So we are not sure that it's the beginning of the stress. And second, you propose to select the performance of the specific THI to better fit its stress. And because Clara had a long experience with Maria Carabagno and Manuel Ramon on such kind of case. But I wonder, because if we change, if we don't fit on Legendre polynomials or with different polynomials, maybe it can be interesting.
We did it and it didn't change that much, because I think what is important is to have the gradient of temperature. So as far as you have that, it doesn't really matter. And on the top of that, if you have the other strain, you can also estimate the stress. So you have the whole range. Physiologically, it makes more sense. So, but in any case, we look at so many different things. We got the data, the only thing we did in the end that is sure is that we impose, when we edited the data, we impose to have a number of observation and heat stress. If that is for sure, but basically the only thing that changed. And I think I remember having that this discussion. Yeah.
Sorry, because
And of course, they have only 1.5%, which is a different.
for example, if we not sure what, because it's a bit hard for the genetic part for fertility, but I wonder this question. If we focus on this part, because here the polynomials for us, as there is an increase at the beginning and at the end, it's better to fit another two.
I'm thinking trying to keep things simple in the model. Because
Exactly.
all these polynomials, if we go to different the results and in the end.
When you do the same, if you apply the broken line.
Exactly. But
But you give-
with us, the broken line is a problem because it's clearly not stable.
No, but in
Clearly
the
not stable after
in
before
the
the
broken
threshold.
line, in the broken line, what you do is
It can be better
Yeah.
for this one.
What advice is to do?
we did and we use the broken line and I realized that my represented also the broken line.
Due to lunch, I think we have to close here. We can still discuss that during lunch, of course, or after.
I'm very sorry for the time.
No, it was
I'm very sorry.
Thank you for the presentation. Yeah.